Hello, good evening, everyone. Good evening, good evening, everyone. Guten Abend, auch gut bei dir. So you have a lot of questions today. So I have opened up many links for you on top uh, to solve each question one by one because it's very hard to answer your questions singly, everyone. I can answer to your questions if you ask me your questions in yes or no. Uh, that will be easy for me, but long questions with long explanations, and especially on typing, it's a cumbersome job actually, to be honest. I received very nice emails also with your uh, assignments, your queries, uh, many queries about project, how should we go. I will share my PhD with you. Then we go for it. Uh, there was a question. There was a very nice email. I don't know who sent me that email. There was a long email I wanted to reply that email, but I thought I will reply to everyone today in the evening. Yeah, this one, I guess. Uh, so I would be happy if you make your points in clear and print point, like you're not just asking me things for the sake of asking. But if you want to ask your query, somebody has to change for Zoom. So we are trying to get Zoom also now, to make things better for you, um, so that we can have live sessions face to face again. Um, yeah, so we are sorry if my voice is too pitch is height, or I don't know, is it low pitch? Uh, I'm a bit sick, I guess. I don't know. Tired, I would say, from the long day. So, tell your friends to join if somebody is nearby you. Uh, so I see you at 8 then. So let's wait for 10 minutes more, then we start. Um, I'm really excited. I'm really excited for you today. Uh, as in the presentations that I have made for all of you. Um, they are quite important ones. Sm- pres- Today we might be finished early, but if somebody's, I think today I cannot speak even louder than this. But I will try my best. I will try my best. Um, just check. Okay. Um, yes, students. Please come by. Please come by. Very good evening, everyone. Very good evening. Welcome to the class, third day. More to learn, more things to go for you. Um, so most of your problems were with the project related, how to write reviews, and everyone seems that they want to write a review on their own. For being group, it's quite difficult. My voice is low. Um, because I'm speaking slow, that's why it is low. I Let me get something to make the voice louder. Let me just come by. Just stay there. I will come within 3 or 4 minutes. Then no, thank you so much. Um, if my voice is not loud enough for you, then the session will be not good enough. It will be not easy to understand. But with the flow, we will be faster. It's yeah. Don't worry, I will be louder. Don't worry, don't worry. Uh, uh, yeah. So today agenda is so I wrote on my notebook because of various questions. So what I did, I summarized questions in my mind so that I can answer all together. So one agenda would be your review, various examples, how research paper is made, how to write your own paper, and how you can publish in free journal. Second agenda is your like to ask about going abroad to study in Germany. Third agenda is our okay, we are here to study first of all. So yeah, molecular biology methods that we're gonna do today, and also I will add some to applied field of molecular biology to it little bit with medicinal biochemistry and proteomics part if we have time today. So we will have mixture of everything. Do we have touch of molecular biology, proteomics, INPCN, your your proteomics part. I hope you know proteomics. How many of you know what is the definition of proteomics? Please write down in the comments. Proteomics and medicinal biochemistry. Wow, very nice definitions coming up there. Uh, Proteomics has imperative are imperative pieces of living beings with numerous capacities. The proteome is the whole arrangement of protein that is delivered or altered by a life form or framework. Oh, nice one. Proteomics is the study of protein structure and how it functions to a specific stimulus. Hmm. Medical biochemistry deals with enzymes. It's about how a drug interacts with various cycles in our body and their enzymes. It's also deal with the metabolism of the drug. Yeah, you're quite coming quite near to the point. Yes. सर citations के लिए कितने minimum पढ़ने बच्चे I will explain how many papers you have to study. You can uh, read as much as you want. Um, we will discuss this point because if you understand the publication pattern, how publication is done. Let me add my hard disk to computer today so that I can show you my PhD data. How uh, why I said PhD is like going to a Mount Everest. Um, I'm attaching my uh, hard disk to computer today. And the one, she's my sister. Okay, doctorate, miscellaneous. Uh, so that was my writing time of my PhD. Uh, so let it be 8 o'clock or more. Let more people add. Then it will be nice to discuss things with you. Uh-huh. Uh, Let me find out my dissertation also to you. So if somebody is interested to read my dissertation, how a dissertation, your PhD thesis is made, it's online available on the Göttingen website. Yeah, there. If you just copy and paste my status of uh, so these were my professors, my referees. It's 29 MB file. It's okay. In India, internet speed is fine. It's not that bad like earlier. In Germany, we used to have like 50 Mbps, quite common in our house. Uh, Here's is also fine. It's good enough. Just wait a second. Hello, ma'am. Yes, ma'am. गुड इवनिंग. हाँ मैम, थोड़ा सा ना मैं करता हूँ कुछ. हाँ. ठीक है. नहीं वही आज वही ठीक है मैम. ठीक है. So, very good evening, everyone. Uh, I have my three, good evening, everyone. So, I have three agendas for today. That is review a little bit about studying abroad, like where to go, and about our topic, what we have to study today. So, I will start. I will start now. Okay. So, let me start with you first of all, how to write your review actually. So please, I will count to 20. Please bring your pen and copy. Okay? I will start counting now. And within this 20 seconds, get your copy. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. I hope you all have your copies with you and pen. Yeah? I am speaking louder. What's happened today? So, voice is better? Or still, uh, voice is very low. Why is it so? For some it's low, for some it's okay. So if my voice is not good, it will be not make sense actually. Hello, mic testing, mic testing. One, two, three, one, two, three. It's better. Yes, it's clear. Yes, yes. Thumbs up. Okay. Yeah, I had already my earphones with me. Too much. Decrease your phone voice down then. Um, let's start with a review thing. Yeah. So if everyone know about PubMed and CBI website, if not, I will start with the basic because some people know, some don't know. So what you have to do, just uh, like on PMC, when you go to the NCBI, just write down these names: ncbi.nlm.nih dot government. Yeah, and there you just write whatever the topic you are looking for. Whether you're looking for DNA microarrays, gene therapy, cancer cell line, or any other thing, you can check it here. Okay. Um, then let's say if I write DNA microarrays, so this is the first review that came, and on the right hand side, what you can see, it's a different where you can open it. It's pub reader mode, EPUB mode, PDF mode. So best is the PDF mode. Just click on the PDF mode afterwards, and there you go. You have your one review already written by someone, and just see the pattern how it has been written. First is the name title, yeah, then is the name of the person, then comes the abstract, a short summary of everything, uh, then the keywords of your review, then comes the overview. First of all, the introduction. So this is how they have divided in their case. They started to talk about early history of DNA arrays to start with the history. Yeah. Then the birth and of the modern DNA arrays. Spotted arrays, various type of arrays has been talking about. Okay. So then self-assembled array. So within them, they are talking from the past to the present, what has been done in the past and what is in present. That's the main reason of review. What people has done in past and what you think you could think of in the future you could do. You can also add your scientific ideas in them. So that's the main reason to bring the past to the people, like what is has happened in the past, what is happening in now in the present, and what could be happen in the future. So bringing three different time zones together, so and bringing that knowledge to up to us. Okay, then comes the applications. So first we discuss various types, the history, then comes the applications, various applications, under them various sections are there. This is one example. This is how somebody wrote their review. So you can see various trillion, zillions amount of reviews. Then future of DNA RS, as I discussed. So you can write what could be the future, what are the things that are happening to make the bring out the new techniques in the future in the within that field. Then at the end, acknowledgement to the people who have motivated you to do so, and then literature cited reference should be in always in same order like all have. You can use some softwares like EndNote or some other reference preferred softwares, uh, reference library softwares. So like in this case the reference has been made like all the last names first, then the first names are in small letters, then the comes your title of the topic, then comes the year, then comes the page number, then comes the yeah, I think this was the volume number and that's the page number. So it's difference in different way of doing it, and PubMed code is there. So that's how you can make an even reference your references. So I hope it's clear how to write your own reference letter. So I can share this link to you now on the Viber. This was one example. This is not Viber. Oh, and this is not Viber also. This is Telegram. I always forget that. Uh-huh. Okay, just download it, have a look over it. Uh, so what we will do today, we will have last 10 to 15 minutes for your queries related to your projects, your questions, and we try to answer them as much as possible so that you don't have to write me personally or write me via an email, and we can discuss things here only. Yeah, that will be better thing for us. So, I mean, about the page number of review, uh, you can write around, let's say, 3 to 4 pages or 5 pages. So the content needs to be good. That's the main part. But the main reason to do these exercises are that you understand how the academia works. So it's not that you have to be really good, really perfect, but it's just that you taste the academia, how academia works, likes how each thing that you are doing, you're thinking, you're writing, you make thinking, your thoughts are being penned down. They make a lot of sense and they make a lot of heavy weight to the audience. So that's when you're writing your presentation, your sorry review. Just think over it. And I will also discuss little about dissertations, how dissertations looks like. Though you don't have to write, but somebody might be writing their master thesis or PhD thesis. For them, it might be useful. So this is the link. If you want to check how a PhD thesis prototypes looks like. So I can share this here also again. And also onto this one so that we do not forget to share things. Oh, I forgot in this. So I will share in both groups actually, whatever we are studying today. I think this one, this one, this one, this one. Yeah. So, okay, good. Um, So review part is over. I hope many doubts are cleared. And yeah, if somebody wants to write review on their own, you're most welcome. But if you can write in a group, that's also most welcome. Make a group of 2 or 3 people. That will be nice actually. And if somebody is not part of the project, they didn't pay it, if still they are interested to write review, they can still write it. We will most welcome it, but you might not get the certificate. For that, you have to ask Vatsal sir and Shivani ma'am how to go for it. Beyond that, somebody's is not able to type the thing, so you can send it to me via writing, via clicking on the phone, with your pen also, with the assignments things. Okay. Now the second agenda. I hope it's clear with your review part. Uh, if there's any question with the review, please write it down now. Pages, so bache, pages, it's up to you. It's like you're writing your own story. You're writing your own Harry Potter. So this could be a 7 series Harry Potter. So you can write a 7 series review also with 1,000 pages in each review. No, it's up to you. I will not stop it to write to you. So you can write maximum as you want. Minimum at least 5 pages will be good. At least 5 pages, so that you have a feeling of how good writing has to be done. Yeah. People in the group, it's all you're open actually, because I felt some people, they are more willing to do their own things, they have their own ideas, their own pattern of thinking, and they doesn't mix. But I am extremely saying this to make group, because that's where you learn. Because wherever you work in a group, in a science field, you have to work in a team. And if you start to understand other person's likes and dislikes, and you try to compromise with, or you can also what do you say, alternate the other person's liking dislikings, if you have that power, superpower, I would say, you can go for it. But that's how the teamwork, you can learn in it. Yeah. There are many things as the team working works. Team management uh, helps a lot for your working in future. So try to merge with the groups. That's recommendable. If you can't, then go for on your own also. That's also fine. Everyone whosoever, after that 10 days are over, within 3 days or 4 days, you have to send it your projects to us. Yeah. And maybe you can send it on my email, or you can send it on the um, we can create one group on the Viber for sharing only your projects. Yeah. So I will talk about that. So about project sharing, or we can make some Google Docs. Somebody suggested me. So we will see what will be the best, so that you all can see each other's file also, and you can learn out of it. That would be better. That was a very good advice by someone. Okay. Any other questions? So my last 10 seconds to for this. Any other questions about reviews? Yeah, about publication. So guys, I can help to you to some extent for publications, but there are many free journals available. uh, Free journals to publish in what field? Okay, molecular biology. So you will have list of journals there: Allen publication, Journal of Molecular Biology, and contact the editor directly. That within the internship, virtual internship that happened with Edufabrica, we had this chance to do a review project, and we would like to publish that. This is our format, how it looks like. Just write one email, simple email to them. Don't send your everything at once. Just ask them whether they can help you in or not, for your free. Sorry for that. How you can do it. So how to find free to publish journals. So there are many links are coming up. Just go through them and look for it. So just it's all about how you tackle Google. You can find your solutions with that. Okay, so that's it for from that point of view. Um, so second thing is now. About your topics, because many people were asking me to think about abroad. So I will talk about little bit. uh, जाओ, जाओ, just wait a second. Sorry guys, it's just my sister. Uh, okay, so review part is over. So somebody is interested with where I did my masters. That's the link for that. University of Gießen, for the masters in agrobiotechnology. So I will share the links just now so that we don't forget them later. So I'm highly active now at the moment for all of you. Uh, so there you can see the details how you have to go via UniAssist. You have to send your application via UniAssist and get it done. So you can get it done, and it's all free. It's a public university. You have to pay just 400 to 500 euros for one semester. Okay, and it's a 4 semester course. Started in 2007 accreditation they got it. So you have to just pay to give TOEFL test to get at least 550 points on paper based test or 80 points on internet based test. IELTS is minimum score is 6 is required in the academic test. So that's the minimum requirement to study there. And yeah, just ask on this email ID for the application if you have any questions. They are very, very helpful to you. And yeah, so these are the core modules that you will be studying there. Special biochemistry 2, biostats, bioinformatics, plant protection and bioengineering, biotechnology and genomics, microbial food biotechnology, molecular phytopathology, risk assessment, molecular entomology, industrial internship. So believe me, these topics that what I had studied there, they were very to the mark, and a very good way of teaching was there. The professors were very nice. Of course, you have European life also, European food also. Things got changed for you. So that was something a little bit about my university for my masters. If you're interested, go for it. And about job offers for PhD people, these are the Fraunhofer's Institute. There you can just Google it with the career part there, jobs and careers, where they have many different groups of working in different fields. And it's a huge in Germany. Everywhere there are big groups in there. So you can have a look in there also about them. Fraunhofer Institutes. Yeah. And also one more, Max Planck. So these there you can check in the career if somebody is looking for PhD type. So mpg.a, because these things are very in front of us, but we just neglect them because no one is there to told us about what is the future about science, how you can have an extra. Uh, Sorry guys, I have to do it now because later I will forget to it. Then it will be not nice for you. Yeah, and if somebody's looking for masters, this is the website for that, mastersportal.com. There you can search for any course and any country actually. But for Germany, you can just like if somebody is interested for structural biology. Okay, there is no course as structural biology. Anyhow, biotechnology, and you look for country Germany. Oops, double N, nanny. So you will have huge amount of universities name coming up there. You can look for them and apply whichever you feel the best. So there are 26 universities that came that they are teaching in Germany. Also Sweden is also a nice country to do so. But Germany I'm preferring because it's free of cost. You don't have to pay anything. Okay. Then comes the DAD. This is the one of the best website also. This is from the Germany itself. Please go through this website also if you are looking for PhD or masters. So this is just not only subjective where I'm teaching you, but this is also career counseling is happening so that you can take care of your future. So we are spending quite time on this, but it's good. Somewhere you could be benefited out of it, of my experience from my life. So another point is I have created one quite quite number of months before, new career consultant. There we are just talking with each other and discussing things about going abroad, studying abroad, or studying in India, about various examinations, CSIR, NET, JEE. So it's a good platform to discuss with each other wherever you are looking for. So just come in, join in, and share your thoughts. Share your things. What do you think? I can also answer some questions there. Uh, so just to be in touch with each one of you, you can join this group later. And one or more last thing, that's kind of advertisement now, I have to say. Uh, it's my brother's wife, my bhabiji. They had started their own immigration after having 15 years experience in this immigration for sending students to the Canada, to Germany, to everywhere. So being in personal touch, if you, I can refer to you, all of you. They can, whatever the fees will be they are charging, they can reduce it. So you can look for this UV immigration. They are in Chandigarh, Sector 22. We had started our own immigration consultancy recently after having a 20 years of experience. So I am just um, yeah. So they have already sent a lot of students to Germany, so they are good in the paperwork, which I might be not as they are good. They are professionals actually, so I will prefer them. If you have any questions anywhere, anything, and please, I humbly uh, request you. Uh, I've also posted one video here. Don't get trapped into the people of false consultancies. So there's one video, why education consultants promote private universities. This is one of the YouTubers there. He published this because uh, the private universities pays uh, in India, they pay for they pay to the consultant people to tell them to go to the private university, and they charge a lot from you. Yeah, they charge a lot from you, and in that case, and you pay a lot in the Germany also. They will say if you get pay investment, if you do the investment, you have to get a return also afterwards. So but that's not true. Germany is free of studies. Don't think about that. You can just go to the public universities. Don't go for private universities. And any questions left for this point of view? It's already 30 minutes we have spent on our review task and broad task questions. Um and about if any another questions you have, please you can ask. You have 1 to 2 minutes windows open for that. So we will not take any further questions on that. So I want to solve these things. That's why I created this time for you, to bring everything, this agenda for you, so that we can solve everything just like that. So everyone questions will be solved in this. Yeah. So there are many also scholarships available for PhDs and masters. Yeah. German in Germany, yes, it's good point to learn German. If you have done, if you at least before going there, you do a A1 level, and then after staying there, you can continue studying that to the C1 level. That's good if you look for future there, if you want to stay there for longer time and looking for PR. So for that, the German is very necessary. There is no word limit for project. That's okay. So it's 25. I start at 27 then. If you have any other questions, please you can ask regarding the review and studies. Don't worry, this is your first time. People who are writing this review for the first time, I can understand. When I wrote my first master thesis, it took me around 2 months to do so. It was quite hard for me also, but at then it's a success. It is a success. So don't worry. For publishing review, as I explained already, that I can help you to which review should go. I can do that for you, and you have to write an email to the editor by your own, that can you accept our paper? Can you we have this thing, and can you accept this for us? So you have to do it on your own, because it's somehow you need some professors who are authorized to the university. Even this, you are also authorized to, because at the moment I'm not authorized to any universities. So I was part of from a university, but due to COVID situation, we were not, yeah, not allowed to join there. But this is how it is. This is the fact. Uh, get your professors with you from university and tell them that you want to publish one review, you have written that, and go with your professors. I would recommend you. Your professor will be also happy if they can see that you are doing something on your own and you want to publish as a review, so they can also help you to get it done. Yeah, because you need some authorization from university while publishing. Okay, it's 27, so no more questions now. I have to start now teaching you. So just let me remove my hard disk. Okay. So today I will be dealing with you mainly the PCR, RT-PCR, DNA microarrays, plasma cloning, next generation sequencing, and knockout mouse, and also a little bit about CRISPR-Cas9. So these will be the 5 to 6 topics that we'll be dealing in detail today, with a lot of videos, presentation, and yeah, no, not this one. So I shared you the review format, I guess. I will just repeat it. So it has to have title page, title, on the title page, your title, your name, date, then on an abstract, it should contain around 200 to 300 words, summary of what you think of your primary study, reviewed, and conclusion of the study. Okay? And then comes your introduction of your clearly outline the order in which subtopics are discussed. Then comes the body. This includes your subtopics that you're thinking for. Then comes the conclusion, then literature cited. So just keep in that mind. So day 3. So this is our agenda for today for our techniques in molecular biology, third day. Welcome to the class. So today I will be teaching you about analysis of nucleic acids, how you can analyze the nucleic acids by a PCR, gel electrophoresis, blotting techniques. So these two things we will do on separate day. Gel electrophoresis, I think on the 6th day. On the 5th day is the protein analysis. On the 7th day, I guess, is the blotting techniques. Northern, southern, western, eastern, northern, southern, many others. Then gene expression analysis we will do today, real-time PCR, with which we check our COVID-19 situation, we check our COVID-19 patients. Microarrays, that's quite exciting thing. Then we check the cloning by plasmids with this thing called recombinant DNA technology. Then we discuss a little bit about Sanger sequencing and the next generation sequencing like Illumina, Iron Torrent, all those ones. Then comes the method to study gene function, cell culture methods, and generation of transgenic mouse and knockout mouse. So that will be our agenda for today for our third day. So these are the various yeah, DNA microarrays figures there. So uh, a cat being uh, green fluorescent protein GFP being green here, So these are the differentiated cells, embryonic differentiated cells. Here you can see growth hormone. With the growth hormone, the mouse has size has been increased, and this is your NGS, next generation sequencing. With still we are using by Sanger. This is for your example of showing you northern blotting, how northern blotting, southern blotting has been done. This is a plant of Arabidopsis thaliana as a model plant of plants to show you things, how things are working. Okay, so first thing first, PCR. Um, so what do you think about what is PCR is? What is your views on your PCR? What do you think about PCR? Tell me. Mm-hmm. Okay. To increase the amount of DNA, yes. Technique to amplify DNA, yes. Helps in the amplification of gene of interest through various cycles. Yes, that's quite nice definition. True, true. So we will make your complicated process quite easy today for you. So we will do a virtual lab for the same. So some people have done this with me earlier in the virtual lab of biochemistry, but today you have to face it again. Sorry guys, but enjoy that time also with us. Okay, so I got good answers from you. I'm very happy. I will make a small clap for you. Clap, clap. So nice. So polymerase chain reaction, it's mainly means amplification of DNA. Here we are just amplifying the DNA for our various applications. So it can be used to amplify a rare specific DNA sequence from a complex mixture when the end of sequence are known. We just need to know the ends of the sequences and we can increase our sequence to whatever level you want. And in this amplification, mutant alleles allows detection of the human genetic diseases. We can check the human genetic diseases by checking by increasing the mutant alleles to be in higher amount. And beyond that, DNA sequence can be amplified by PCR for using in cloning as a probes and in forensic science. So for the same technique, Carey Mullis got the Nobel Prize winner in chemistry in 1993. Um, so and while doing so, so you can see on the right hand side there is a three different cycles have been shown to you. Cycle 1, cycle 2, which yields 4 molecules, cycle 3 yields 8 molecules. Let's start with the first part. So this is your genomic DNA, like here 5 prime to 3 prime and here 5 prime to 3 prime, and our target sequence is this much. We want this much part of the target sequence to be amplified, only this much. So first denaturation will happen with the heat. We will separate the DNA strands like that. Then annealing will happen. We will cool the temperature a bit down and let the primers to form the hydrogen bonds with the target of sequence. They will move to the end of the sequence primers by decreasing the temperature. Then what will happen? Extension will start with the help of DNA polymerase enzymes, and we will add some nucleotides to it, which go from 3 prime end to each primer like this. Okay, and hence you have nucleotides to be added there. And then further, this was your first cycle. So here we also increase a little bit of temperature. So here's the temperature, we decrease it, then increase it a bit. And then this was your first cycle. Like this, we have one set of our DNA molecule, and then the same cycle will repeat with these different parts. So we'll have four molecules of DNA molecules here. Then again cycle 3, 8 will happen, and like this it will continue. So in the PCR mixture, in the reaction mixture, we add buffer, template DNA, primers, nucleotides, DNTP as we call them, DNA polymerase, and water. So this is the PCR mixture you created in a small epi in your tube. So what? Just wait a second. Okay. So for after this, I will take you to the virtual lab now. I think this one was there. Yeah, PCR. Let me just have a good size for you. So human genome is made up of 3 billion chemical base pairs. Scientists often need to isolate a specific segment of DNA from within a vast amount of genetic material. And this we can do with the help of PCR. So here you can see the cell nucleus, DNA. So polymerase chain reactions can generate 100 billion, yeah, 100 billion of identical copies of specific DNA sequence in matter of hours, in a few matters of hours. As the inventor of PCR explained, this requires no more than a test tube, few simple reagents, and source of heat. That's it. Three things that we required while doing PCR: test tube, few simple reagents, and source of heat. That's it. So this is your hair follicles. So to check PCR, you need DNA that has been extracted from the cells. So that's the purpose of PCR. So we make DNA copies from from your source, and because initially we have very less DNA, and that's not enough for your further reactions. So you can do with the help of a sample of blood, skin, saliva, or even hair follicles. We can choose any part of them for our DNA. So let's start this virtual lab for us. Uh, or start by moving your extracted DNA into your special PCR tube. So this is everything is done here on the ice. So you can see there's a basket there. There's a thermocol based basket. So in that we have ice. In the ice we have these epi tube stands here with the small one here. So let's start the thing. So let's drag the extracted DNA. So like we did the isolation of DNA, remember? Did we did isolation of DNA virtual lab so far? Did we did on the first day or not? Can you just tell me with yes or no? Did we did the DNA isolation in the virtual lab or not? We did. Okay, great, 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 great. So you see that let's say we have extracted that DNA that's to the PCR tube. So we just zappened off and we put into the PCR tube here. So this is your PCR tube at the moment. So we just add DNA to that. Now we will add primer 1 to the PCR tube, and primer will attach to the DNA strands at the end of the segments. We want to see the copy. So they are the powerful tools for copying your specific DNA sequences, and they do not target the wrong sites also. So here we have primer 1. Now we will add primer 2, which will attach to the second site. Okay, we throw it away. Now we will add nucleotides so that our reaction could go further. A, T, C, G. So we will add all DNA codes to it. Our nucleotides will be added. Okay. Now DNA polymerase, final thing will be added, DNA polymerase to make the reaction go for further. Okay, now PCR tube is filled with all reaction components, and now we will put it in the DNA thermal cycler, which will change the different temperature. It will heat and cool our tubes, and these changes will be the crucial for making our reaction work. So this is our thermocycler. So we put this in our Appendorf into that and start. So here you see what happened inside. That's the magic. So this is our target sequence. So we have a temperature of 95 degrees Celsius our inside our PCR tube. And at this temperature is like almost boiling. So at this temperature, DNA double helix start to separate from each other, creating a single strand molecule. Okay. Then we decrease the temperature to 50 degrees Celsius, suddenly half of that, and then our primer 1 and primer 2 get attached to the different ends of our DNA. Then we increase the temperature in the third at the 72 degrees Celsius where our DNA polymerase is attached, and also we are having our DNA nucleotides. They will attach and DNA polymerase will help to go further directions, and DNA nucleotides will be keep attaching to it. So like this. So this was one cycle. We have our target DNA sequence that we are looking for. So we again make the temperature 95, then 50, then 72, then again like this. We will continue like that. So this is your desired fragment at the end. After 3 cycle, you have your desired fragment with 2 DNA molecules. So we will continue these things for like number of cycles. This is cycle 4. So you have 8. After 4 cycles, you have 8. After 5 cycles, you have 22 for fragments. So we will continue like that. We will keep continuing like that. So after 30 cycles, you have a huge amount of the same sequence. You can have it there. So this is how you're within 30 cycles, you're with the help of some reaction tubes, one Appendorf and one these and the thermocycler. So you have created a huge amount of DNA with help of them. Okay, all good? Understood PCR? Any questions? PCR is good? Today got understand with it? Please thumbs up if you understand today all about PCR. All good students? Okay, understood? So there is a lagging, that's why you reply late. Okay. Okay, great. So let's continue. Uh-huh.